We're rolling.
We decided to do our presentation project on the SMN1 overexpression as a potential treatment for spinal muscular atrophy. What is spinal muscular atrophy? Spinal muscular atrophy is a genetic disorder, more specifically an autosomal recessive neurodegenerative disorder caused by a mutation in both copies of the SMN1 gene on chromosome FQ. The SMN2 gene is unaffected in those with spinal muscular atrophy. Spinal muscular atrophy is characterized by the weakness and loss of skeletal muscles caused by the loss of motor neurons that control motor movement. SMA affects about 1 in 11,000 births, or 1 in 50 Americans. And 1 in 50 Americans is believed to be a genetic carrier. What are the SMN1 and SMN2 genes? Humans have 2 genes for the production of the SMN protein, SMN1 and SMN2. The SMN1 stands for survival motor neuron 1 gene, produces the functional SMN proteins, which are very important to maintain the health and the normal function of motor neurons. The SMN2 gene produces mostly, but not entirely, non-functional SMN proteins that cannot be used to maintain the normal function of motor neurons. In individuals with SMA, the mutated SMN1 gene leads to a decreased production of this protein, of the SMN protein. Without the proper levels of the protein, motor neurons in the spinal cord will be lost, preventing the muscles from receiving the proper signals from the brain. Spinal muscular atrophy and the SMN genes. In those unaffected, those who do not have SMA, the SMN1 gene produces just fine the functional SMN proteins that are needed, but in those who are suffering from SMA, the SMN1 gene does not produce those functional SMN proteins, but the SMN2 gene works just fine, but unfortunately doesn't produce enough of those functional SMN proteins. In those who do not suffer from spinal muscular atrophy, electrical signals travel along the neuron's axon, branching through the muscle and connecting to individual muscle fibers at the neuromuscular junction. And that's how they communicate with, the neuron communicates with the muscles. However, in those who have muscular atrophy, that neuron can't make that connection at the neuromuscular junction because the muscle is so atrophied. And it leads to this gradual decrease in the mass and strength of the muscles over time. Since patients with SMA inherit both copies of the mutated SMN1 that cannot produce any functional SMN proteins, of the SMN1 gene. So what is overexpression? Overexpression is the process by which the biological yield, in this case proteins, is produced at a much higher frequency than normal by the insertion of RNA, or in this case, RNA or DNA. So since patients with SMA inherit 2 copies of the SMN1 gene that cannot produce any functional SMN proteins, we decided that we would look at doing an overexpression of that gene. In a recent study done on mice, it was found that the overexpression of the functional SMN1 gene was able to delay the onset of spinal muscular atrophy. SMN1 overexpression may be a possible therapy for SMA. However, the downsides of the overexpression of the gene have not yet been explored.
Okay, so to look more into this topic and kind of base our experiment off of, we looked at an article titled Neuron-specific knockdown of the SMN1 causes neuron degeneration and death through an apoptotic mechanism. So what the researchers did in this experiment was they used C. elegans to examine this gene, and they looked into the specific neuron, the D-type motor neuron, which has an RNAi transgene. And the job of this motor neuron in the C. elegans specifically is kind of how they are able to move backwards. So the scientists looked at the young C. elegans and looked at their backward movement specifically, since that's what the neuron does. And to test this function, they would first touch the nose of the nematode and see how it moved backwards. And they would also touch the end of the nematode to see how it moved forward to compare. And they would score the movement of the nematode. And a defective movement would be if they were unable to move backwards and correctly move backwards. And then the percentage of the defective movements of the animals were looked into, and the percentage was calculated. Okay, so through this research, this is what they found. The knockdown of this SMN1 in these neurons caused defects in the backward movement, which is kind of what they expected to see. The normal backward movement was greatly reduced. It was about 27% had the normal backward movement compared to 95% of normal backward movement in normal C. elegans. And they also found that the forward movement was not affected, which is also what they expected to see. So that was good. Okay, this is their first figure. So as you can see in the first graph, figure A, SMN1 is the shortest bar in black, and it had significantly less backward movement than both of the controls. The negative control is the KAL1, which is just a different type of gene that was knocked down, and the control is just regular C. elegans. And 60 C. elegans were observed for the control, and 120 were observed for the negative control, and 120 were also observed for the SMN1. And these are the, sorry, can you go back? Thanks. The percentage of the backward movement that they calculated, as I mentioned in the last slide, is what they're measuring here. And the bottom pictures are pictures of C. elegans that are expressing GFP in their ventral cords, and GFP is just a fluorescent protein, so that's just showing their ventral cord.
So our study question is there any drawbacks to the overexpression of the SM1 gene? Because it's already been proven to be able to help rescue degenerating neurons who have SMA, and to also help slow the onset of SMA. So when the SMN protein is expressed 2 to 3 times higher. So the question is there any drawbacks to the overexpression of the SM1 gene? Does this harm the organism in any way? Does it consume too many raw materials that are not present in the in its food source? Does it require a specialized diet for that? Does the protein interfere with any other biological process within the organism? If it's too over concentrated of that protein, does it get anywhere else and then mess with other neurons and other proteins? Is there any adverse effect on the motor neurons? Does the overproduction of the SM1 gene consume parts of the different proteins within the neuron so that it can be produced? Or is it fine? Or is the overexpression something that is perfectly fine and acceptable for the organism? Overview of the experiment. So we're going to be using C. elegans as the model organisms. They will be containing the human gene of SMN1 instead of the SM1 gene, or similar gene that does a similar thing for the motor neurons. The lock-on system will be inserted upstream of the SM1 gene, and this is how we will control the expression of the gene. The SMN1 will be controlled by the concentration of lactose present in the C. elegans medium. The C. elegans will be divided into 4 groups. We will have the negative control, which will have the mutated C. elegans containing no lactose. So if in this control, if the C. elegans develop SMA, then that will be correct. That will be what we are looking for in that one. Then we have the control, which is just normal everything. Normal C. elegans and a normal medium. So they should live perfectly fine with no problems. The positive control are going to be mutated C. elegans in a petri dish that contains enough lactose for the normal expression of the SM1 gene. So that way, that will be our comparison to the negative and to the control. The positive control should look like the control, is the hope. The experimental group will contain mutated C. elegans in a petri dish containing an extremely large concentration of lactose. This concentration should equal to 5 to 6 times the actual concentration. This will produce 5 to 6 times the concentration of SM1 protein than in normal, C. elegans. So, a quick explanation of the lac-operon system. So, in the lac-operon, the system of the genes is just a set of genes that have promoter and repressor regions. Lactose is a promoter for this system, meaning that all the genes that are downstream of this will have a higher chance of being transcribed. So that's simple. So, and then the model organism, of course, we chose C. elegans. Now, why did we choose this? It is a widely used model organism that has been used to study just genetic disorders for many years. It has been used to study motor neurons before. They are easy to maintain, easy to study. The motor function is easily seen and quantified. They've been done for a number of years, and they have had a number of human genes inserted into them before to be used for study in different genetic disorders. So they are a very good choice to use and very cost efficient as well. And materials we, of course, will be needing is petri dishes, C. elegans themselves. We will be needing the mutated C. elegans. We will be needing the normal C. elegans. We're going to need the mediums for them, concentrations of lactose, microscopes, and timers, and of course a few other things, but those are the main things of interest. And the procedure. So what we'll be doing is we'll be doing multiple petri dishes for each group, just to ensure that if anything happens to the C. elegans, anything that gets on them, that can end up killing them, we have extra. And if there's any data that seems to be wrong, we can at least find out what the problem is and compare it to the rest and look at the outliers. So we'll have 10 C. elegans from each petri dish. We'll have the movements observed qualitatively, so that's just to see are they moving in a way that is what we would consider normal, in this case, thrashing back and forth, eating the food in a straight line, and comparing it to the known models of how they move. The lengths of the C. elegans will be measured along with its width, the width at its midpoint. The idea here is to see if the C. elegans at any point are smaller or thicker than they should be, then we can tell because maybe the neurons are being destroyed and not moving as much, and as such they are smaller. That's a good way to tell. The C. elegans will also be placed in a drop of water, and we will record the number of times they thrash every 30 seconds. This will be able to determine if the motor neurons that they have are being slowed or at the same speed or have anything affected to them. They should move. The number of times they thrash in 30 seconds should be similar between them.
Okay, so our anticipated results and the conclusions that we came to, we anticipate that the experimental C. elegans on the plates that have a lot of lactose will thrash more than each of the control plates, and they will show increased movement, and that the experimental C. elegans will also be longer and be larger than the control C. elegans. So if our anticipated results are true, this could potentially lead to a better treatment or a new treatment for spinal muscular atrophy. And this treatment may not have as many physical downsides and financial downsides as the other treatments out there have. So we're hoping this can possibly improve the lives of people with this condition.
And then our references. So thank you for our presentation. We hope you have a good weekend.